Imagine being on a space station, orbiting hundreds of kilometers above Earth, and discovering a life form that Earth has never seen before. In a recent breakthrough, Chinese scientists aboard the Tiangong Space Station collected microbial samples from their habitat, and when those samples returned to Earth, they revealed something extraordinary, a brand new bacterium species that may forever change how we think about survival in space. The bacterium is called Niallia Tiangongensis, named after the space station where it was found. It's not entirely alien. It's a close relative of Niallia circulans, a bacterium commonly found in the soil. But the Tiangong strain is unique. After being exposed to the harsh microgravity and radiation conditions of space, it seems to have evolved new traits that make it stronger, tougher, and more resistant to damage than anything we've ever documented in this genus before. This discovery was part of China's CHAMP program, the China Space Station Microbiome Project, which aims to monitor microbial life in the closed environment of crewed spacecraft. In May 2023, the astronauts of Shenzhou 15 took samples from the walls and equipment aboard the Tiangong station. When scientists on Earth cultured and sequenced these microbes, one strain stood out, JL1B1071T. Now formally classified as Niallia Tiangongensis, this bacterium is a scientific marvel. What makes it so special isn't just that it survived in orbit, it's how it adapted. This microbe forms incredibly robust biofilms, allowing it to cling to surfaces and resist stress. Its spores are unusually resilient, and they act like biological armor in extreme conditions. Most fascinating of all, genetic analysis revealed that Niallia Tiangongensis has enhanced DNA repair capabilities. Specific proteins involved in mending damaged DNA, like ligase D and SPLA, show variations that could help it rapidly fix damage caused by high levels of radiation. This is crucial in space, where cosmic rays constantly bombard living cells. The bacterium also displayed a rare ability to hydrolyze gelatin, a trait not typically seen in its Earth-based cousins. This suggests it may have adapted to consume different nutrient sources, potentially allowing it to survive even in nutrient-poor environments. These biochemical quirks hint at a broader metabolic flexibility, making it a strong candidate for biotechnological applications in future missions. Now, let's talk about what this means. First, for astronaut health, knowing how microbes behave in space helps researchers manage biohazards and prevent unwanted contamination. If a bacterium forms tough biofilms, it could pose risks by sticking to air filters or sensitive equipment. Understanding it is the first step to managing it. Second, it opens the door to new science. If we can understand how Niallia Tiangongensis resists radiation and repairs DNA, we might apply these principles in medicine, perhaps developing better treatments for radiation exposure, or even designing organisms that clean up nuclear waste. These mechanisms could also be useful in developing probiotics or skin protection products for space travelers. Lastly, this discovery fuels the ongoing question in astrobiology. If life can evolve and adapt like this in a space station's artificial environment, what might be possible on the Moon, on Mars, or in the subsurface oceans of Europa? Niallia tiangongensis is a small organism, but it carries big implications. It reminds us that life is astonishingly resilient, and that space is not empty. It's an active frontier where biology continues to surprise us. The more we explore beyond Earth, the more we realize that survival out there may not be as impossible as we once thought. This bacterium isn't just a passenger in space, it's a pioneer,